talking about, it's actually a continuation of my previous class where we talked about the development, embryonic as well as post-embryonic development of sea star. And if you recall, this is the life cycle that I'm talking about right now, that this release of gametes and then early embryonic development leading up to the formation of gastrula, and then some more development of gastrula and transformation into this early larval form, which develops into this bipinnaria stage, and then it is transformed into brachyolaria stage. So these two larvae are of sea star, and also in turn these are the larvae of asteroidea, class asteroidea, that includes sea stars. So then later this part here, it is containing the rudiment of the future sea star. So this part breaks off from this, and then further development occurs. This is a young juvenile starfish. It's less than 1 mm, and then it grows, and several systems then grow into these arms, and it matures when its reproductive system is functioning into adult male and female, and the cycle starts all over again. Now, in this particular slide, we talked about these two larvae, that is the bipinnaria larva and the brachyolaria larva, these two larvae, right? So that means these two larvae are commonly present in the sea stars. That means in the class Asteroidea, right? So today we will be talking about other larval forms belonging to other classes or all the classes of echinoderm, phylum Echinodermata. So these are various larval forms. There are diverse larval forms present in phylum Echinodermata. So some of them are these which are common, such as bipinnaria, brachyolaria, Ophiopluteus here, Echinopluteus, this one. I hope this diagram is visible. I'm just magnifying and zoom in it a little bit here. So this is Auricularia and Doliolaria. And if you recall the name of classes, there is Ophiuroidea class, Echinoidea class, Holothuriidea class, Crinoidea. So in these different classes, there occur different larval forms, right? Some intermediate forms also develop that we may just mention in passing, but these are the main larval forms that we will be talking about. So let's start with, we have talked about bipinnaria and brachyolaria in great detail. Here you see in this bipinnaria, several lobes appear and a primitive gut is formed here. Just follow the cursor here. This is the primitive gut, and not that differentiated, having mouth and some precursor of stomach and intestine and anus, so straight alimentary canal, because it is just developing at this stage, and some formation of these lobe-like structures and these ciliated band around it. So this is the early stage. Then later these arms develop, and we have talked about the names of these arms. Some are on the dorsal side, so they are dorsal, some on the median, and on the dorsal side they will be called as dorsal median arms. Some will be on the lateral side, so these will be lateral arms, anterolateral, posterolateral if they are on the posterior side, ventral side. If it is present above the mouth, that would be preoral. It is below that, that would be forming the postoral. So these are, according to their location on the larval body, these arms are named. So in this, just lobes are formed, and here these arms are formed. So these two are asteroid larvae. Then we have Ophiopluteus. Now this is the larva of Ophiuroids. Ophiuroids contain, if you recall from my first class, there are some echinoderms that have snake-like arms, which start at the narrow junction, and they are extended like that. Not like starfish or sea star, where arms start broadly and then taper. In the ophiurites, arms are or almost entire at the entire length. They are of almost uniform this thing thickness. So this is the larva. Ophiopluteus larva is of the ophiuroids. Brittle stars भी कहते हैं उनको commonly ophiuroids को. Then echinopluteus is of echinoids, as the name indicates. Here you see it is having these forwardly directed arms. Here also you see these forwardly directed arms. Here the arms are like more dispersed, whereas in this case the arms are more compact, and they are of almost entire similar length. Here you see these two arms are more developed. They are elongated. These are the longest ones. And then we have auricularia larva, which somewhat is similar to this bipinnaria larva. Here you see auricularia, and then we have this doliolaria larva. This is of the crinoidea class, and this is of the holothuroidea, sea cucumbers. And the crinoids include sea lilies and feather stars, etc. So in different classes, we have different larval forms that we will be talking about in detail today. So let's start with the echinopluteus one here. We have talked about these two in detail. So this echinopluteus, this again forms like this bipinnaria. It forms after gastrulation. We have studied in detail how gastrula is formed. First, there is zygote formation that restores two in number when both male and female gametes unite. A zygote formation occurs, then successive cell divisions and during cleavage, and then solid ball of cells, that is morula, then blastula, where a cavity is formed and cells are arranged peripherally, and then invagination occurs and primitive gut or archintron forms. That part is gastrulation. And after gastrulation, this echinopluteus larval form appears. And here the gastrula becomes conical, and the gut is differentiated into mouth, esophagus, and stomach, etc. Similarly, as we studied in the previous, the bipinnaria one. Blastopore forms anus. We know that this is a deuterostome. Then larva forms projections, which develop into arms. Now here we will be talking about what arms are present in this case. So there are preoral arms, anterolateral, anterodorsal, then postoral, posterodorsal, and posterolateral. So if you look at this, there is a preoral and a postoral. There is anterolateral, there is anterolateral and posterolateral, and anterodorsal, posterodorsal. So that way you can just imagine where they are located. Some are on the dorsal side and anterior, some are on the dorsal side and are posterior. Likewise, some are on this lateral side anteriorly, while others on the anterior lateral side posteriorly. So depending on their location on the larval body, these names are given to different arms. So several arms are there. Now, the tip of arms here are pigmented. Tips are pigmented, and they are supported by calcareous skeletal rods that keep the arms stiff. Of course, the locomotion is by ciliated bands. So ciliated bands appear, which becomes thickened. These ciliated bands become thickened, and they are called as epaulets. So the location where these ciliated bands are present on the larval body, that portion is called as epaulets. So let's look at this. So these ciliated bands, you see these structures, these small bands, they appear on the body, and they are called as epaulets. All these are epaulets. They are ciliated bands. And then various arms, some are labeled here, some are not in this one. And you see this part is the gut, the primitive gut, the larval gut. And here during development, this is the echinus rudiment, just like you saw in the starfish rudiment. in the brachiolaria one. Here in this case, you see this is the echinoproteus rudiment, which later gives rise to the whole echinus. ठीक है, जो sea urchin होता है. And then these are different arms, the post-oral, the anterolateral, the pre-oral, etc. And this central part is the gut, the alimentary canal, and not that well differentiated gut. It is a primitive sort of gut. Later, when adult is forming, then this whole alimentary canal is restructured. The internal development, here you saw the externally this is formed. Internal development also goes like bipinnaria. Here the archentron gives off the hydroenterocoel, that is a coelom is formed, and then this is an enterocoelic animal. You know very well what enterocoelic animal is, what are enterocoelic organisms. Then the coelom develops and is differentiated into three parts: the exocoel, the hydrocoel, the somatocoel. We have already discussed in case of bipinnaria. So this particular coelom finally forms exocoel, hydrocoel, and somatocoel. Then an ectodermal invagination on the left side. The hydrocoel gives off 5 radial arms and 5 primary podia. And the lantern, you know that in the echinus, there is a typical characteristic feeding apparatus that we call as Aristotle lantern, which I mentioned and showed that structure in my some previous lecture. So that lantern is formed from left somatocoel. Then you can this and other different areas give rise to, some develop more while others they just are show the reduced development and give rise to various systems like water vascular system. Let's go back to this one. Here you see in case of C star. So these are 3 coelom differentiated into 3 parts: the somatocoel, the hydrocoel, and the exocoel. This part further develops on the left side, and this is disintegrated here. And then you see this ring formation occurs. This water ring develops, and this is further develops. Here you see this water ring. So the coelom contributes to the formation of these channels which will later form the water vascular system. So similarly, such type of development occurs in case of other larvae like echinoproteus. Then echinoproteus is, we don't need to go into much detail of embryonic development of all the larvae, but we just want to know the basics about this development. So since lantern is a very prominent structure in case of echinoids, so here we have mentioned that it is formed from left somatocele. And this echinoproteus larva is a microscopic structure, and it's a free-swimming larva, and it develops in 7 to 30 days. Then metamorphosis is rapid. It takes place in about an hour, and there is no attachment stage in echinoids. If you recall from the previous one here, this is the attachment stage. These are the adhesive discs with the help of which the larva is attached to some substratum. And further development from this to this stage occurs in the attached condition. And later the stalk breaks, and this is then is released, and it's free, and then it develops. So bipinnaria, early and late, is free swimming. Then this it develops arms, and these arms have, some of these arms have adhesive discs. So now it spends the attached life. Some development occur during that condition, and then this breaks. There is a break, and then this part containing the rudiment of the future star fish is released, and then development occurs. So in case of echinoids, in case of echinoproteus, there is no attachment stage in this. So there is this larva, the echinopluteus larva, and the development of larva occurs in this pattern. And metamorphosis is rapid, that is change from the echinopluteus to the juvenile, and then growth occurs in the juvenile. So let's come back to this here. This is the like kind of 3D picture of echinopluteus. So arms are of comparable length in this case. Now Ophiopluteus, here this is again a free swimming larva of brittle star. Sorry, we were talking about echinopluteus. So echinus ka hai, sea urchin, right? And then we want to talk about the brittle star one. Okay? So Ophiopluteus is of brittle star. This is a free swimming larva of brittle star. And it is similar to echinopluteus of echinoids. But Ophiopluteus has fewer arms than echinopluteus. Fewer arms compared to the echinopluteus. The posterolateral arms are the longest and they are directed forward. Here we are talking about these arms. Here, these arms are longest ones and they are directed forward. The arms develop after gastrulation, as we have seen in the echinopluteus one. The posterolateral arms are formed, they formed first. After 4 days, anterolateral arms develop. After 10 days, postoral arms develop. After 18 days, posterodorsal arm develop. So this is the pattern of development of various arms in the larva. And all the while, the internal organs are also developing. Then ciliated bands are there on the arms edges, and the internal development is same as steroids. That is how the coelom differentiates and then differentiates into exo, hydro, and somatocele, and some part of exohydro give rise to a particular organ system, and so on. So internal development is similar as that of steroids, and finally this larval form appears here. You see, the name is like this. This is the posterolateral. Then we have posterodorsal arms, anterolateral arms here, and this is the primitive, the larval gut here. You see, and the two number is this is left hydrocele with lobes. We know what hydrocele is. This is a part of coelom. So this is Ophiopluteus larva. Now you have to remember the names, the structure, and what part of digestive system has formed in these larvae, because these are also included in your practical exercise as well. So in practical exercise, you have to make this diagram and you have to write few points about various arms. This is again Ophiopluteus, the same picture that we have seen here. This is Ophiopluteus, this is the real picture, this is the diagrammatic one, and this one is the real picture of the Ophiopluteus larva. All these are microscopic. Then auricularia larva, it is a larva of holothuroid. If you remember the classification slides, there are sea cucumbers having a cucumber-like body, not a body like sea star or the globe shape in case of echinoids, or long arms like the brittle stars, ophiuroids mein jaise. So here the body is elongated and cucumber shaped, and auricularia is the larva of holothurids. So you have to remember the larval form of a particular class. ठीक है ना? Auricularia जो है वो किसका लार्वा है, या बाइपिनेरिया किसका लार्वा है, या ब्रैक्यूलेरिया किसका है, या ओफियो वाले कौन से हैं, ओफियोप्लूटियस किसका है, या इकाइनोप्लूटियस किसका है, right? So this is the holothuroid larva, After gastrulation, we know that up to gastrulation, the things are like the development proceeds in a same manner. That is cell division, morula formation, then blastula formation, then gastrula formation. And after gastrulation, the formation of coelomic sac occurs and the gut starts differentiating. Coelomic sac, that is the exocoel, the hydrocele, the somatocele, etc. At this stage, the embryo becomes free-swimming larva called as auricularia larva. It is transparent, pelagic, and 1 mm. It swims about by a ciliated band, which forms the preoral loop and the anal loop. Internally, it has a curved gut and hydrocele and right and left somatocele. So let's look at this. Here you see, this is the gut, larval gut, and we have this part hydrocele, and this number 5 you see the somatocele. राइट साइड की है, लेफ्ट साइड की सोमेटोसील, हाइड्रोसील इस साइड, एक साइड में ज्यादा डेवलप है, दूसरी साइड में ये डिसइंटीग्रेट हो रही है, एंड इट इज that further give rise to different organ systems. The gut is it's centrally placed here. There is a larval mouth, larval anus, and these are the ciliated bands or lobes present. So in a way, if you compare it, it looks like somewhat looks like our which larva? Bipinnaria larva. So here you see this is the auricularia and this is the bipinnaria larva. They are somewhat similar, whereas Ophiopluteus and Echinopluteus, they are somewhat similar here. Let's come back to this auricularia one. So it swims about by a ciliated band which forms these loops. Internally it has curved gut and the hydrocele and left and right somatocele. So this is the structure we were talking about. Here you see this is a beautiful picture of this larva, auricularia larva. And you see several lobes and the gut is visible here, and these ciliated bands, preoral and the preanal and the dorsal side and on the ventral, lateral sides, these lobes are, here in this case, the lobes are more pronounced compared to our bipinnaria larva of C star. Here you see. So this is a kind of give you an idea of a 3D structure. Okay, it is folded like this. It is having the flap like structure. Okay, ये side view आप देख रहे हैं. तो कुछ structure आपको dorsal side पे दिखते हैं, कुछ structures आपको ventral side पे, then lateral. So depend करता है where these lobes are located. You name them accordingly. And here you see the cilia. So this is it's a electron microscopic figure. SEM की picture है ये, scanning electron microscopic figure, and you see these ciliated bands which help in swimming and procuring food as well. Then there is a transitional doliolaria larva, right? It is a transitional stage from auricularia. Here, in some cases, this auricularia larva transforms into doliolaria and then into the sea cucumber, while in others it directly develops into the sea cucumber. So in some cases this transitional doliolaria stage occurs. It is a transitional stage from auricularia. It is barrel shaped and it looks like doliolaria larva of crinoids. Crinoid कौन से हैं? Sea lilies में. उसमें उसका जो अपना लार्वा है that is doliolaria larva. लेकिन होलोथोरॉइड्स में भी पहले कौन सा लार्वा बना? Auricularia, and then it transforms into transitional doliolaria. And this transitional doliolaria larva resembles the typical barrel-shaped larva of crinoids. Crinoids जो है वो एक क्लास है, एक क्लास में आने वाले ऑर्गेनिजम्स को कहते हैं crinoids. ठीक है, crinoidea के अंदर. So looks like doliolaria of crinoids. There are continuous ciliated bands. They break in 3 to 5 flagellated rings, right? So earlier there were continuous bands, then they form 3 to 5 flagellated rings. Then metamorphosis is gradual, during which it acquires tentacles and podia. Sometimes this is known as pentactula larva. When it is developing these tentacles and podia, then that is called as pentactula larva. In one holothuroid, that is in Cucumeria, this is the generic name, this is an example of holothuroid, there is no auricularia stage, and embryo directly develops into doliolaria. Right? So there is a huge diversity in terms of development also. So mostly there is a auricularia larva, but in Cucumeria there is no auricularia stage. The embryo directly develops into this stage, the doliolaria stage. In Holothuria, this one, embryo directly hatches into a young, no larval stage. So in some cases, in this particular class also, there is direct development. In case of asteroidea also, in some cases, there is direct development, which in which case the no larval stages appear. So this is the transitional doliolaria larva. Here you see this becomes barrel shaped. इसमें देखिए there are these lobes present, but in this case it is a barrel shaped, and these bands of cilia, they are there. So they are differentiated into patches of these ciliary structure, ciliary bands are there, and the openings, one forming mouth and other forming anus and so on. This is again a SEM picture, the scanning electron microscopic picture. Next we come to the doliolaria of crinoids. डोलियोलैरिया ऑफ द क्रीनोइड्स, ठीक है। क्रीनोइडिया एक और क्लास है जो कि कंसीडर करी जाती है स्टॉक्ड, हालांकि उसमें कुछ फ्री स्विमिंग भी हैं. दैट दिस क्लास इंक्लूड्स these feather stars and sea lilies, etc. So here this doliolaria larva is of that class. It hatches as free swimming larva. Body has 4 to 5 ciliated bands. There is an apically sensory plate having cilia. There is an adhesive pit over the first ciliated band. Then between first and second band is present stomodium or vestibule, and the skeleton develops. Stomodium makes up the part of foregut, that is mouth, pharynx, and esophagus. After differentiation of organs, larva attaches itself, and internal organs rotate at an angle of 90 degrees from ventral to posterior position. Larva forms a stalk, and now it is called as pentacrinoid larva. Pentacrinoid. Crinoidea ka hai, forms a stalk. We know that several of these crinoids are stalked animals. Okay? So next the larva forms a stalk, and now it is called as pentacrinoid larva, and metamorphose after some time into adult. So this is the barrel-shaped larva of crinoid, the doliolaria larva, जिसके बारे में हम अभी बात कर रहे थे. Barrel की तरह है, and it is having ciliary bands. There is an apical tuft of hair also. This is 4-day post-fertilization. ठीक है, fertilization होने के 4th day पर ये इस फॉर्म में डेवलप होता है. That means all the stages have already passed. That is the morula, the blastula, the gastrula, and then this is transformed into this doliolaria larva. This is the detailed structure. Here you see there are ciliary bands, skeletal plates develop, there is this adhesive pit, and the sensory tuft here. So this is the detailed structure, and it hatches as a free swimming larva. Body has 4 to 5 ciliated bands with apical sensory plate at the anterior end. It is provided with bunch of cilia. Here you have seen in this picture. There is an adhesive pit over the first ciliated band near the apical plate in the mid-ventral line. Then between second and third ciliated band lie the stomodium or vestibule. Skeleton develops at this larval stage, and after differentiation into different prospective organs, the larva attaches itself, and the internal organs rotate at an angle of 90 degrees from ventral to posterior position, and the larva forms a stalk. Then it is referred to as pentacrinoid larva, which metamorphose into adult. So this is what we talked about. These are some more pictures to give you an idea about these larval forms. Now, what is the significance of these larval forms? So, since all these are bilaterally symmetrical, that give you an idea about the common origin of classes, except the larvae of crinoids, which become sedentary. Other larvae have some fundamental resemblance. So crinoid का कौन सा larva है? This one. This becomes sedentary after the stage because it will give rise to sedentary adults. Construction, they are constructed on the same fundamental plan, having bilateral symmetry. Now we are talking about all these larval forms, that is bipinnaria, brachiolaria, oviopluteus, echinopluteus, doliolaria, etc. So that indicates towards the origin of classes from a common ancestor, which was coelomate, bilateral, and free swimming. Then these diplurula and pentactula are two such hypothetical ancestors, which millions of years ago they appeared, and then they gave rise to this line of development and these echinoderms. And all these modern echinoderms have originated from these ancestors, which are named as diplurula and pentactula. Then the importance of larvae is that they aid in dispersal and feeding. Since adult echinoderms are somewhat sluggish, they don't disperse that much. Their larvae are the main dispersive phase. They remain in this planktonic stage for sufficient time to disperse far from the place of their birth to new areas, finding different food sources. So what is the purpose of this dispersal? So that the larvae and adult will not compete with each other for food and space, etc. So they aid in dispersal and feeding. So this is another significance of these larval forms. The larvae form is evidence that this phyla has evolved radial symmetry from a bilaterally symmetrical ancestor. Right? If you recall from the classification scheme, we have at very early stage we have these bilaterally symmetrical phyla. ठीक है, उससे पहले रेडियल सिमेट्री आती है, किस फाइलम में आती है, यू सर्च दैट. ठीक है ना, ये एक क्योंकि आप और अर्ली फाइला भी पढ़ रहे हैं, you must be knowing about that. So just recall which phylum has this radial symmetry. So there is one offshoot that give rise to that phylum. ठीक है, मैं एक हिंट आपको दे देती हूँ, इसमें मेडुसा वगैरह फॉर्म. Right? Then after that, the animals are bilaterally symmetrical. And then again, from those bilaterally symmetrical ancestral forms, this again, there is another line that has this radial symmetry. So this radially symmetrical animals have probably formed, or definitely that they have formed from a bilateral ancestor. And this larval form is used for dispersal that we have talked about. Now, in this one slide, you see all these larval forms. Here, you see this one, the early stage, a hypothetical diplurula larva, then bilateral stage of pentactula, then pentactula after torsion here, then we have here bipinnaria, we have brachiolaria, then we have E is the brachiolaria larva, then F is the auricularia larva here. This is auricularia. So you see this somewhat resembles this one, bipinnaria. Then we have at G point, we have transitional stage from auricularia to doliolaria. Okay, this one is the doliolaria. Okay, so this transitions from this stage to this stage and then to this stage in some of the species. of holothuroids. Then we have echinopluteus and we have ophiopluteus. So this is echinopluteus here. And you see the orientation of these arms in case of echinopluteus and here in case of ophiopluteus. Ophiopluteus brittle star का है और echinopluteus sea urchin वगैरह का, sand dollars का है, जो कि थोड़े अडल्ट स्टेज में कॉम्पैक्ट ऑर्गेनिज्म होते हैं. उनके आर्म्स फ्री आर्म्स नहीं होते हैं, आर्म सारे मिलके एक बॉल या एक ग्लोब की फॉर्म बन जाती है. सो दिस इज अवर इकाइनोप्लूटियस एंड दिस इज of the brittle star, that is the ophiopluteus, snake like arms. Ophio word designates snakes. Then we have doliolaria here, and then late doliolaria larva, and this is the pentacrinoid stage here, pentacrinoid stage, where this stalk has developed and the attachment plate has developed, and then this gives rise to the sea lilies and feather stars, etc. So this is of the stalked ones. So this is the general, like it's in this figure you see all different larval forms and how do they look like. Now this is as the animal get more advanced, they have more specialized features than those animals found in the prior phyla. So here we see the single-celled ancestors, then multicellularity over the course of evolution was achieved, and then this sponges, this one branch formed sponges, then tissue grade of organization was there, and then the radial symmetry developed here at this point in this offshoot, and this nidarians developed, while the other shoot that continued three germ layers and the bilateral symmetry. Bilateral symmetry continued up to here. So bilateral symmetry and protostome development, that is the blastopore giving rise to mouth. So this is the common feature up to here until a deuterostome development occur at this point. Flat worms, then another shoot is of round worms having pseudocoelom, and then at this point a true coelom developed. True coelom has formed. First pseudocoelomate came and then the true coelom developed, and then these are the true coelomate and protostomes, the mollusk, annelids and arthropods. So there is a common ancestor at this point, then two branches, one giving rise to mollusca and other giving rise collectively to annelid and arthropods. And then at some point in evolutionary trend, these two branches formed. One gave rise to annelids, current day extant annelids, and the arthropods. And then deuterostome development, where the blastopode giving rise to anus. So this is the common ancestry for both echinoderms and the chordates. Then this part became the radial symmetry. This branch gave rise to radially symmetrical animal. So in course of evolution, this radial symmetry has evolved independently at 2 points. Then these, of course, are chordates and then chordate classes, or pisces and amphibians and reptile and so on. And some protochordates, जो कि अब सबको phylum का status है इस वक्त. Maybe you will be studying about those in your next semester, that is animal diversity 2, how further classes formed, what is the peculiar features of various classes at this point. So that was the phylogenetic development of various phyla here. Now in this we see phylum echinodermata. So this is the common stalk, and then one crinoids developed, these stalked ones, and another branch gave rise to all these four. So all these four are closely related compared to the organisms or the species present in the crinoids. Then from this common stalk, these two branches developed. One gave rise to asteroids, the sea stars, and another branch gave rise to all these three, that is ophiuroids, echinoids, and holothuroids. And then this branch gave out these two forms, the holothuroids and echinoids. So here you see these two larvae, they are somewhat similar. They are similar. And here you see the asteroid and the holothuroids, they are somewhat similar. Okay? At the larval stage, they are somewhat similar. So they look that they are closer, but this is not the case. right? They are very distant in their phylogenetic scheme of things. Now here you see, this is the deuterostome evolution. Here you see ancestral deuterostomes, and then two branches originate. One gave rise to phylum Chordata and other to phylum echinodermata. And then various classes have formed here. So this is in another scheme. You see that then this offshoot becomes separated of tunicates and then cephalochordates, and then the one branch gave rise to these vertebrates. So the larval symmetry always does not indicate the taxonomic affinity among the Elythrozoa. Now, if you recall that in one scheme of classification, we have formed two subphyla. One was Erythrozoa and other was Palmatozoa. So different people in different times have considered different characteristics and classified echinoderms. Right? So in a like older scheme of classification, Elythrozoa and Palmatozoa were the two subphyla. So among the Elythrozoa, two well-marked larval forms occur, that is Pluteus group and Auricularia group. Pluteus is common to Ophiuroids and the echinoids. Pluteus, ठीक है ना? अभी हमने देखा कि Ophiopluteus है और Echinopluteus है. They are having bilaterally symmetrical, all are actually bilaterally symmetrical, but there are long arms present. And auricularia is common to asteroids. It resembles bipinnaria and holothurians. ये भी हमने देखा कि auricularia जो है वो asteroids के bipinnaria को resemble करता है और holothroid. So on the basis of larval similarity, Ophiuroids should be placed near to echinoids because their larvae resemble, and asteroids near to holothurians. But this is not in agreement to paleontological and morphological studies, according to which the asteroids and Ophiuroids are closely related, and echinoids followed the independent evolution. तो हालांकि उनके लार्वा सिमिलर हैं, बट अडल्ट में वो divergence दिख रही है. Right? So there is close larval similarity in Ophiopluteus and Echinopluteus. This is an example of convergent larval evolution. Occurrence of convergence is seen among the unrelated groups such as Asteroidea, Holothroidea, and Crinoidea. So there is convergence here as well as here. On the other hand, larvae of closely related forms such as asteroids and Ophiuroids exhibit major differences, and which must be due to divergent larval evolution. Therefore, the larval structures in echinoderms cannot serve the purpose of determining phylogenetic affinities. So this is their taxonomic significance. Auricularia of echinodermata and Tornaria of these protochordate balanoglasses show very close similarity. This as well as cleavage pattern, the mesoderm, the enterocoelic condition indicate that echinoderm and chordates are phylogenetically related groups. Okay? Because Tornaria is in the protochordate. So यहाँ भी similarity है protochordates में और echinoderms में. So that means both are phylogenetically related groups. And this is again another way we see this scheme of evolution. Here you see that this is the showing the development of radial symmetrical adult echinoderm from bilaterally symmetrical larvae. So these are the adult Ophiuroidea, Echinoidea, Holothroidea, Asteroidea, and these are the larval forms. This is again a picture of SAM, and here you see these larval forms, different larval forms, the bipinnaria and the developing stages of Ophiopluteus. Just go through this. At this point, it is not that visible, but this is a good slide, and go through this slide. You will be following this phylogenetic development of various classes and various sub-phyla, what are the common features and what are the specialized features, how these specialized features kept on adding and giving rise to various branches that ultimately formed different classes, developed into different classes. So with this, I finish this, and I will be sharing this PPT on my Google Classroom. We'll be continuing our Monday class onwards. अभी भी जब तक आपका लास्ट वर्किंग डे नहीं आता, हम क्लास कंटिन्यू करते रहेंगे, हम चीज़ें डिस्कस करते रहेंगे, आपसे फिर 1-to-1 डिस्कशन होगा. So with this, I end my syllabus, my part, my share of syllabus here. But all of you have to join class on Monday. Then we will be discussing about some of these topics again. If you have some issues, some problems, we can discuss that. I will be sharing this whole PPT on my Google Classroom. So please visit that. Tell your friends who are not attending the class to visit my Google Classroom and look for these PPTs. ठीक है. एक मैंने और शेयर किया था आपसे सेशनल के बारे में. आप सेशनल की डेट्स बता दी हैं, टॉपिक दे दिए हैं, शॉर्ट नोट्स लिखने हैं, बहुत क्रिस्प. ऐसा नहीं हो कि 20-20 पेज उठा के आप जमा करते हैं. बिल्कुल टू द पॉइंट एंड क्रिस्प सेशनल आपका होगा ये. या आप इसको वो भी कह सकते हैं कंटिन्यूअस असेसमेंट का पार्ट, या आप इसको असाइनमेंट भी कह सकते हैं. तो ये तीन
अप्रॉक्स कितने पेज हो सकते हैं?
पेज में तो मैं इसलिए नहीं कह सकती, जैसे आप क्लासिक जनरल कैरेक्टर वाला अगर क्वेश्चन अटेम्प्ट करते हैं तो वो तो 2 पेज में हो जाएगा. ठीक है ना.
यस मैम, उसमें पेज कम होगा.
हाँ हाँ. तो पेज को तो हम इस पे नहीं कर सकते हैं, लेकिन हाँ मतलब 400 वर्ड्स के अराउंड कितने पेजेस होंगे उसमें. डिपेंड करता है, आप हैंड रिटन करेंगे तो ज्यादा पेज हो जाएंगे, टाइप करेंगे तो कम हो जाएंगे, या आप वर्ड के फॉर्मेट में दे रहे हैं. हाँ तो लेकिन बीच बीच में आपको फिगर्स भी लगानी होंगी. आप दोनों को कंबाइन करके भी कर सकते हैं कि पिक्चर्स आप वो वाली लगा दें और टेक्स्ट आप लिख दें, या आप टेक्स्ट हैंड रिटन दे दें, पिक्चर्स आप वैसे ले लें. तो आपके ऊपर है सब कुछ. लाइक देयर इज कंप्लीट फ्रीडम. लेकिन पढ़ के लिखिएगा, अच्छी तरह से एक्सट्रैक्ट करके लिखिएगा. ऐसे तो आपको पीडीएफ मिल जाएंगे, आपने उसे उठाया, कट पेस्ट किया, लेकिन वो कट पेस्ट वाली चीज नहीं. जस्ट गो थ्रू द टॉपिक एंड देन राइट दीज शॉर्ट नोट्स. एक्चुअली शॉर्ट होने चाहिए. 6 नंबर में आप इमेजिन कर सकते हैं कि कितना हमें रिक्वायर्ड होगा. तो सब अच्छे टॉपिक्स हैं, इजी टॉपिक्स हैं, और जब बुक से पढ़ेंगे तो आपको अच्छे से समझ में भी आएगा. ठीक है.
And we have to make some diagram also.
Yeah, diagram are must in zoology. ठीक है ना. बट लेकिन आप क्लासिफिकेशन अगर बता रहे हैं, आपकी मर्जी है. नहीं भी बनाएंगे तो भी चलेगा. आप चाहें तो छोटे छोटे से आपने एक जहाँ पे आप शो कर रहे हैं कि फॉर एग्जाम्पल कि कोई एक पर्टिकुलर फीचर तो एक हैंड ड्रॉन सा बना सकते हैं. नहीं तो नहीं भी बनाएंगे तो भी कोई इशू नहीं है उसमें, इन केस ऑफ क्लासिफिकेशन जनरल कैरेक्टर वाले में. Right? But when you are talking about general character, you have to give some examples of both these phyla. ठीक. और कोई issue?
No ma'am.
जैसे मैम मोलस्क है जैसे, तो उसमें जनरल कैरेक्टर के बाद डायग्राम्स अगर एक बना दें और 2 एग्जांपल्स तो. यस.
हाँ, एग्जाम्पल तो देने ही आपको.
यस मैम.
मैम लीव कर सकते हैं क्या?
हाँ बिल्कुल. I am going to leave the class now.